Okay, so welcome to part 3, the final part of these 3 videos introducing you to physiological principles. In this one, for some of you, it will be revision, but we're going to go over the cell and give you an overview of the cell. So the learning outcomes for this final section. First, we're going to look at the nucleus and the contents of the nucleus, which of course you remember the nucleic acids from section 2, where we talked about DNA and RNA. We're going to look at the structure of the plasma membrane, which again, if you remember from section 2, is composed of phospholipids in a bilayer. So it is a phospholipid bilayer that surrounds the cell. We're going to look at the cytosol and the cytoskeleton. The cytoskeleton is the scaffolding within cells, the cytosol being the fluid. Together with the organelles, this forms the cytoplasm. We're going to look at the structures and the functions of the endoplasmic reticulum, the Golgi apparatus and the mitochondria. And these organelles are all membranous structures. They are constructed using the same phospholipid bilayer as the plasma membrane. Finally, we're going to look at ATP, adenosine triphosphate, universally known as ATP. And this is our source of energy for cellular activities, and it's synthesized via cellular respiration. So animal cells are eukaryotic and are mostly nucleate. In other words, they have a nucleus. They have evolved beyond the primitive anucleate, without a nucleus, prokaryotic cells. Prokaryotes have no nucleus, no real chromosomes, and no mitochondria. Bacteria are prokaryotic, but they can replicate. In other words, they are anucleate. They don't have a nucleus, but they can still replicate. And this replication in terms of bacteria and infection may be called colonization. Viruses are not even cells because they cannot replicate independently. They have very simple structures with either RNA or DNA, but they lack the ribosomes. Remember ribosomal RNA and the ribosomes? They lack these, so they cannot use their nucleic acids, either RNA or DNA, to replicate. Hence, they need to enter host cells and use the host cell ribosomes. Compare the eukaryote and the prokaryote cells below. Which one is the eukaryote? Yes, it's the one on the left, far more complex. Cells are the structural and functional units of all living organisms. In the multicellular organism like the human, the cells collectively carry out all the processes that make the organism a living entity. Cells vary in shape and size, but in average, most cells are around 10 microns. That's 0.01 millimeters. Because of this, their structure can only be viewed using an electron microscope. And they all start as undifferentiated stem cells. Look at the diagram and identify the morula and the blastocyst. When fertilized, a collection of totipotent stem cells, these are stem cells that can become any type of cell, forms a morula, and this has about 16 cells. Before implantation in the placenta, at around 7 days post-fertilization, the morula hollows out into a blastocyst containing pluripotent stem cells. These pluripotent stem cells can become anything but placental cells. The hematopoietic stem cells, they go to the bone marrow and they eventually become either erythrocytes, so red blood cells, or leukocytes, white blood cells. Others form the cells of the tissues. The replacement of senescent or dying cells occurs throughout the body. Cells can only replicate so many times before they die, and this is a protective mechanism to maintain healthy cells. So all tissues have stem cells to replace the differentiated tissue cells as they naturally age and die. Stem cells can replicate to provide more undifferentiated stem cells. And stem cells can then become differentiated to be what's called progenitor cells. Progenitor cells then fully differentiate to replace specific tissue cells that have died. Have a look at this pathway in the diagram. The generalized cell is composed of 3 distinct regions. The nucleus, which contains the DNA. In the diagram, note the chromatin within the nucleus. The plasma membrane surrounding the cell, and the cytoplasm, which contains all the organelles. Study the picture and identify the organelles outside of the nucleus, but within the plasma membrane. All cells, apart from erythrocytes, remember these are red blood cells, have a nucleus. Skeletal myocytes, myocytes are muscle cells, are the only cell to normally have many nuclei. They are multinucleate. So erythrocytes are anucleate. The nucleus is enclosed in a phospholipid bilayer envelope. This is exactly like the plasma membrane, and this has around 2,000 pores. Large proteins called nucleoporins form the pores, and the pores allow water-soluble molecules to enter and leave the nucleus. Around 1,000 molecules can be transported through each pore every second. And many of these pores communicate directly with the endoplasmic reticulum. The endoplasmic reticulum has membranes that are continuous with the nuclear envelope. In the diagram, find the pores and see how many lead into the endoplasmic reticulum. Also note the nucleolus, the chromatin, and the red dots on the endoplasmic reticulum. The nucleolus then is a dense area within the nucleus where rRNA and ribosomes are synthesized. Remember, rRNA is ribosomal RNA, and it forms the ribosomes that are found on the endoplasmic reticulum outside the nucleus. The ribosomal subunits are the largest structures to pass through those pores in the nuclear envelope. The complex folded polypeptide chains on the right are 2 ribosomal subunits. And these can be seen in our previous diagram as the red dots on the endoplasmic reticulum. Chromatin forms the characteristic chromosomes before replicating to form diploid cells in mitosis or haploid gametes in meiosis. Remember, DNA usually fills the nucleus as spaghetti-like chromatin. So for mitosis and meiosis, the chromatin condenses into chromosomes. The plasma membrane is 6 to 10 nanometers thick. In other words, it's 0.000006 to 0.000010 millimeters thick. In the picture, identify the phosphate heads and lipid tails of the bilayer and the cholesterol rafts that serve to stabilize the lipid tails. Note the glycoproteins and glycolipids on the extracellular surface. Glycolipids are carbohydrate chains attached to a lipid in the outer surface of the membrane. Glycoproteins are carbohydrate chains attached to an integral protein. They act as receptors for binding to hormones, anchors for other cells to attach to, and markers to identify the cell. And they also help to protect the cell. Together, these glycolipids and glycoproteins create the glycocalyx, in other words, the sugar coat of cells. Also note the variety of integral and peripheral proteins. Most plasma membrane proteins are glycoproteins that form channels, receptors, adhesion molecules, junctions, cell markers, or enzymes. Integral proteins have a hydrophobic, in other words, lipophilic, and a hydrophilic region, so that they can either fully cross the membrane, as in transmembrane proteins, or insert partway into the membrane. This is necessary because the inner section of the membrane is lipid, where the tails are, whereas the outer phosphates are bathed in water from the cytosol or the interstitial fluid, and are therefore hydrophilic. Peripheral proteins do not have contact with the lipid tails, so are solely hydrophilic. They like water.
Okay, so let's talk about the plasma membrane of cells. We've talked about phospholipids a bit, but in terms of the plasma membrane, let's put it together. So we have, and I'm sure you've all heard this, that's not a little man, by the way, we have a phospholipid bilayer. So what happens is we have our phosphates at the top, we have lipid tails hanging down, 2 each, and then on the intracellular side we have the same thing but upside down. The big thing about this is the phosphates are hydrophilic. They like water. The lipid is hydrophobic. It doesn't like water. So cell membranes will tend to form a round shape because they don't want to be in contact with the water. The lipids in the middle don't want to be in contact with the water. So we have lipid in the middle. Let's do that slightly different colour. So it shows lipid in the middle. Phosphates there. Hence, they're called phospholipids. If you remember, actually, what they have is they also have glycerol. So we have glycerol, if you remember from part 2, then we have phosphate, because we always have glycerol in lipids, and then we have the 2 tails, the lipid tails. Remember, those are basically chains of fatty acids, just like we had in triglycerides. We only have 2 of them though. So then lipid. On the inside, of course, the cytosol is watery. So we have water. On the outside, the extracellular fluid, the interstitial between cell fluid, is also watery. So we have water on the outside, phosphates next to the water because they're hydrophilic, lipid on the middle because it's hydrophobic. Okay, now in terms of the proteins that we have in the membrane, if we have a transmembrane protein, of course, it has to be hydrophilic on the outside and lipophilic or hydrophobic on the middle. So it has 2 sections. This bit being hydrophobic or lipophilic. Likes fat, doesn't like water. This bit being hydrophilic. So these plasma membrane proteins, if we have integral ones or transmembrane ones, they can form various things. So this, for example, we could have as a channel. So both sides will have to be lipophilic. On the other side, we have more phospholipids. There we go. The phospholipids get scrattier and scrattier the more I have to draw of them. And then another transmembrane protein we could have might be a receptor. This has to go all the way through. So it has to have a section in the middle that's okay with lipids. If we have an integral protein that only goes part way in, again, the same thing. This has to have, so let's take that one out. This little protein here, it's okay with water that side, so it's hydrophilic, but it's hydrophobic, lipophilic on the bit that sticks into the membrane. Okay, so we've got transmembrane proteins, we've got integral proteins, all in the plasma membrane. We have other things that like lipid, cholesterol, because remember cholesterol is a lipid. It's actually a sterol. It's one of those with the 5 carbon rings. And we have cholesterol rafts. The cholesterol rafts are great. What they do is, because this is lipid and it's all wishy-washy, the cholesterol, because of that complex shape, help to form rafts to stabilize the plasma membrane. Okay, so the one we've missed out in terms of plasma membrane proteins is, I'll make him blue because he loves water. Peripheral proteins. So we have peripheral proteins. Of course, they don't need a lipophilic or hydrophobic bit. They're sitting in water, they're quite happy. And often what happens is these are signaling and they might come up and bind to that one. So we have this variety of plasma membrane proteins. They need to have different areas to cope with the plasma membrane. We'll just get rid of our phosphate over there. So these things, we've got various things. We could have, as we said, channels. We've already talked a bit about water channels as well, aquaporins. But they could be ion channels for ions to come through. Receptors. Signaling mechanisms, lots of other things. But some of the other things, and we have thousands and thousands of receptors per cell, but some of the other things we could have are markers. All cells have various markers that tell other cells what sort of cell it is, that identify it. We could have markers, and we could have something called cell adhesion molecules. And these help, these are really cool. These look like a pair of hands, and they hold cells together temporarily, a bit like Velcro. So there's lots of different things embedded in the plasma membrane. And what happens is all of these start to stick out. So it might have lots of little markers, little channels, little proteins, and these are generally glycolipids or glycoproteins. Because they're all they're all carbohydrate, they're all sugars, they give the sugar coat. The sugar coat to all cells that basically makes it look like some sort of fuzzy thing. This equals the glycocalyx. Calyx, coat, glyco for sugar, sugar coat. So your cells are sugar coated.
The cytoplasm includes the cytosol, the cytoskeleton, and the organelles. The cytosol is a clear gel-like liquid inside the cell, intracellular fluid. Note that intracellular means within the cell. Intercellular is between cells, like the interstitial between cell fluid. Extracellular is outside the cells. So this fluid is intracellular, within the cell. It suspends the cytoskeleton and the organelles. And it contains water, ions like K+, Cl-, enzymes, amino acids, and more complex collections of amino acids, protein complexes. The cytoskeleton is the intracellular, within cell, scaffolding, and it's made up of myofilaments, intermediate filaments, and microtubules. The filaments and tubules are basically chains of protein molecules, and they come in various different shapes or configurations. The myofilaments then. Cytoskeletal myofilaments are made of actin arranged in helices. So an actin filament is a helix. It's a protein. The actin proteins form a chain, like a string of beads, and two strings are twisted together to form the helix. Actin interacts with myosin. Myosin is a different protein. They are both contractile filaments found in large quantities in muscle cells. Between them, they move organelles and vesicles around inside the cell. And the movement of pockets or vesicles of structural proteins or chemicals around the cell or to and from the plasma membrane is very important for biological processes. They also allow the cell to change shape and to move around in the extracellular environment. Cells basically can move like an amoeba or a bit like a slug moves. Intermediate filaments especially give stability to cells that have to withstand mechanical stress. They connect the special junctions that hold cells together to form tissues, and we will look at the junctions during the module. Microtubules form short, hollow protein tubes, and these are all put end to end to form these microtubules, the little tubes being called tubulin. They move intracellular organelles, and they also form the centrioles and the spindle during mitosis. It's the spindle that pulls the chromosomes apart for cell division. In cilia and flagella, the microtubules on one side contract, and this is what causes them to bend and waft extracellular things like mucus in a certain direction. The endoplasmic reticulum is a phospholipid bilayer that forms part of the endomembrane, the intracellular membranes. Its structure is exactly the same as the plasma membrane and the nuclear envelope, and it's continuous with the nuclear membrane or envelope. It forms corridors or cisternae that lead away from the nuclear pores, and it comes in two types. Rough endoplasmic reticulum, RER, is closest to the nucleus and is studded with ribosomes. Remember ribosomes and ribosomal RNA? They translate messenger RNA into chains of amino acids that eventually form structural or chemical proteins. Smooth endoplasmic reticulum is where the chains are folded or adapted into specific proteins. So in the smooth ER, SER, the process of forming these polypeptide chains of amino acids into complex proteins begins. The smooth endoplasmic reticulum is also a store for intracellular, within cell, calcium. Calcium double cation, remember. And intracellular calcium has many intracellular functions, such as signaling or contraction. And we'll come across the mechanisms of releasing Ca++ from the smooth endoplasmic reticulum during the module, and we'll also talk about its possible functions. This is a picture of the rough and smooth endoplasmic reticulum. Note on the rough endoplasmic reticulum the little red dots of the ribosomes. And note that it extends out from the nuclear envelope or membrane. Golgi are also membranous organelles, so they're also formed of the phospholipid bilayer. The folded membrane forms into stacks, and they may be between 40 to 100 Golgi stacks per cell. The Golgi receive part processed proteins in vesicles from the endoplasmic reticulum. Look at the picture of one stack and note the vesicles. The proteins in the Golgi are then modified further and repackaged into vesicles, so they may fold the polypeptide chains further, bind them to other chains, or to lipids or carbohydrate to form lipoproteins or glycoproteins. Or they may cleave sections off to activate the polypeptide chains. Because vesicles are just pockets formed of phospholipid bilayer, they can be formed from other membranes or fuse with other membranes. Fusing with the outer plasma membrane of the cell results in exocytosis of the contents of the vesicle into the extracellular, outside the cell environment. So the vesicles leave the Golgi and they can either be stored intracellularly with their proteins or released via the plasma membrane. A mitochondrion, that's the singular term for mitochondria, plural, has two phospholipid bilayer membranes, an inner and an outer. Both of these membranes are the same type of membrane as the plasma membrane and all the other organelles. Look at the picture. The outer membrane enclosing the mitochondrion and the inner membrane create a space between them called the intermembrane space. The space inside the inner membrane is known as the matrix. The folds of the inner membrane create corridors known as cristae. Mitochondria can vary in number from 100 to several thousand within each cell. Because they have their own DNA, they can replicate. Remember Eve theory and mitochondrial DNA from our mothers? Having their own DNA means that the mitochondria can replicate when the cell's need for energy increases. This is because mitochondria synthesize ATP, adenosine triphosphate. Energy is stored in ATP and released for cellular activities. We talked about ATP in part 2.
Okay, so let's talk about those plasma. We've talked about the plasma membrane in terms of around the cell. But within the cell, most or many of the organelles, the little structures, are made of exactly the same membrane, phospholipid bilayer. the key thing about this is that they can then fuse with each other and become part of each other, or come away from one plasma membrane and form something else. So the membranes are sort of interactive. And if we think about the membranes that we have in the cell, we've talked about the nucleus. We've got the nucleus. The nucleus is surrounded by membrane with pores in it. Those pores lead out to the endoplasmic reticulum, little corridors. So we have membrane here, and then the membrane extends out to form this maze of corridors. So we have all these little corridors extending out, interacting, getting really, really complicated outside the cell. We'll finish those off, otherwise we'll run out of board. So we have the nuclear membrane, and we have, in the early part, we have the rough endoplasmic reticulum, because it's studded with ribosomes. Ribosomes made of ribosomal RNA. So we have rough endoplasmic reticulum, and then later on we have smooth endoplasmic reticulum. So RER we can shorten rough endoplasmic reticulum to, and guess what? SER. Okay, so that's our endoplasmic reticulum, our nucleus. Then we have this big pile of membranes called the Golgi stack, or the Golgi apparatus. And here we've got this thing sort of a bit like this. When you start the module, it's good of you to know right before you start, I'm not very good at drawing. So we have the Golgi apparatus. The Golgi apparatus is also full of little corridors. And it's made of plasma membrane, the same phospholipid bilayer. Outside of that, and between that, let's have a slightly different colour, we have other membranous structures called vesicles. So we have the vesicles, we have the Golgi, and then we'll have some more vesicles out here. Vesicles, guess what, are also made of plasma membrane. So we've got another plasma membrane, a bit like the outer plasma membrane, forming a circle. And inside we have stuff, depending what type of vesicle it is. Now these are all sort of membranous. They are also a bit of a commercial enterprise, if you like. Let's have a nice bright colour. Our nucleus and our endoplasmic reticulum is a bit like a factory that makes parts. So this is our factory. There you go. We'll include the endoplasmic reticulum, bless it. Our Golgi apparatus is a bit like our assembly plant. So we make parts, say we make bicycle part. Over here, we're going to put all those bicycle parts together and make a bicycle, and then we're going to store it away until Christmas when someone wants a bicycle. Yeah? So this is our assembly part. Guess what vesicles are? They're our transport mechanism. They're our trucks. Because what they're going to do is carry the proteins that we are making here across. They're going to drive them down the road from the factory to the assembly plant, into the assembly plant, through the assembly plant, various changes happen to them. And out the other side comes another truck that's got our completed bicycle, our completed protein. And that can just sit around and be stored, or the vesicle can fuse with the plasma membrane and be sent out of the cell. So all of these membranes are fusing with each other each time things move from organelle to organelle. Our other membranous organelle, really important one, is our powerhouse. So here we have our mitochondria. Mitochondria have two phospholipid bilayers. They have a double membrane. So you have one on the outside, then you have one squiggly one on the inside, and that gives you a center, the matrix, and it gives you an intermembrane space. So this is our mitochondria. Two lots of phospholipid bilayer to make a mitochondria, and we have thousands of them within the cell. The numbers can vary. The numbers can vary because mitochondria can replicate. They have their own DNA, mitochondrial DNA, and they can replicate. And if you remember, that mitochondrial DNA comes from our mothers. So we all have more of our mother's DNA than we do of our father's. And that's really the most important of the membranous organelles.
Adenosine triphosphate, or ATP, is a molecule of adenosine with 3 phosphate groups, hence triphosphate. Adenosine is adenine and a ribose sugar. Look at the molecular structure. Adenine is a nucleobase made from atoms of carbon, nitrogen, and hydrogen. The other part of adenosine is a pentose sugar, a 5-sided ribose sugar. Bound to adenosine is a sequence of 3 phosphate groups of phosphate and oxygen. The process of oxidation binds the phosphate groups to adenosine with high energy bonds, and this happens during cellular respiration. When the bonds are broken by the addition of water, known as hydrolysis, energy is released for cellular activities.
Okay, so we finished off talking about mitochondria. Now, mitochondria make ATP. They're known as the powerhouses of the cell. So our mitochondria, they make ATP. ATP, we'll talk about the structure in a minute, stands for adenosine triphosphate. Adenosine triphosphate, ATP. ATP, as I said, is synthesized in the mitochondria, and it's used in the cell for lots of metabolic processes for energy. It gives energy. For cellular processes that require some energy. They're like little batteries, if you like. So, okay, in terms of structure, we've got adenosine. Adenosine is something called adenine. And that's then bound to a ribose sugar. Pentagon. Is that a pentagon? It's close enough. Ribose. Yes? This gives us something called adenosine. So that's our adenosine component. The triphosphate comes in, bound to the ribose, where we have 1, 2, 3 for tri-phosphates. Phosphate you'll often see written PI. It stands for inorganic. Doesn't particularly matter. PI. Okay, so we have adenosine and then we have the triphosphate. It's not so much the phosphates that are important. What's important here is the bonds between the phosphates. So if this is ATP, if we take a phosphate off, and the process that does this is hydrolysis. Basically, you add water. Hydrolysis. And that's done usually by an enzyme called ATPase. Ase, anything ending in ase is usually an enzyme. ATPase hydrolyzes, but it hydrolyzes this high energy bond. So this is a high energy bond. And when you hydrolyze it, what happens is you break the bond and you release energy. Of course, you've broken the bond, so no longer do you have ATP. Now, we've got 2 phosphates. So lo and behold, we still have the A. We have ADP, adenosine diphosphate, for 2. Now, we could go on and break this bond too. We could hydrolyze this bond to release more energy. But actually, as we get closer, the energy drops. So this is a really high energy bond. This isn't quite so good. So at this point, our battery's starting to run a bit flat. So we don't get so much energy from that one. Ideally, what we want to do is break this one, release energy, and then we've got ADP and a phosphate, and we can take them back to the mitochondria. So our ADP goes back to the mitochondria. From the mitochondria, after cellular respiration, it comes out again as ATP. And it can be used again. So the mitochondria synthesize ATP that the cell then uses for energy by breaking the high energy bonds by hydrolysis. Thank you for watching all 3 parts of the introductory material for physiological principles. Please make sure that you've understood everything, you've made your notes. before we get to our first taught session of the module. When you do get to the taught session, on the first day, we might just have a quiz.